Moving now to neighbor country of Latvia, Estonia, and we're gonna see the presenter, research professor in Biophysics, University of Tartu, Jörg Pieper, and he gonna tell us insights into solution structures and dynamics of biomolecules provided by neutron scattering techniques. So Jörg, the floor is yours, and we are happy to have you here.
Thank you very much for the nice introduction. I'm very glad to speak at this interesting meeting and to introduce some of our work on structure and dynamics of biomolecules, studied mainly with neutron scattering, but also with X-rays a little bit. This is mainly work done with my postdoc Maxime Golub and me, but to have a short outline, I will have two basic parts of my talk. The first part will deal with solution structures studied by small angle scattering methods, and the second smaller part will deal with molecular dynamics studied by neutron spectroscopy. And at the very beginning, I want to acknowledge, or at least show you, that many people across Europe have been involved in this work. And I also want to thank the scattering centers ILL, MLZ in München, Germany, and FLNT in Dubna, Russia, for providing beam time and for providing support for our experiments. And to come back to the outline for the work on solution structures, I don't want to go into too much theoretical detail, but I will hopefully show you some instructive examples of what you can really do with these low resolution structural methods. And the four basic aspects are studies of oligomerization state of biomolecules, complex formation of biomolecules, then the study of detergent shells or solubilization of membrane proteins, and finally light-induced structures that can, structures that can be induced with external triggers. To introduce the sample systems to you, the first is a protein called Phycocyanin, this nicely donut-shaped structure that you see here, which is a light-harvesting protein in cyanobacteria. The second is so-called photosystem II, which is capable of splitting water into hydrogen and oxygen. So this protein is responsible for the oxygenic atmosphere that we have. But you can imagine that at the moment there is a technological interest in this protein because it may teach us how to produce hydrogen from light. And finally, I will speak about the orange carotenoid protein, which is active in photoprotection of light harvesting systems. Well, you may ask, I show you here very nice high resolution crystal structures. So you could say all the structural questions are solved. Why do we need low resolution methods to study such systems? And there is one basic reason and several particulars that apply to each system individually. But the basic reason is that functional studies on proteins are often performed in solution, in aqueous solution, which is closer to the native state of the sample. So in principle, one needs to prove that crystal structure also applies for a dissolved sample. And if we go into the detail for each of the sample systems that I want to introduce to you, then Phycocyanin has the property to occur in different oligomers, and depending on organism, it may form various supercomplexes, which are not all crystallized, so that low resolution methods can be very instructive for such studies. Then the photosystem II is a membrane protein, and to isolate such a protein for functional studies requires the use of detergent molecules. so that it can be isolated in aqueous solution, which makes it a more complicated problem. And finally, the orange carotenoid protein is very interesting because the crystallized structure applies to the dark inactive state, while the active state has to be induced by light. It's not crystallized at all, so that low resolution methods are very powerful in this case. So what I would like to show you in my presentation is that small angle scattering methods can deliver structures in buffer solution under the same conditions that are used for experiments on protein function, so that structure and function can be directly correlated. But let's start with the first two examples, oligomerization and complex formation. And I use Phycocyanin as an example. You see here it can occur as a hexamer or a trimer. That means if you study your solution by SAXS, you get green points, and advanced simulation tools that are available from the group of Svergun at DESY in Hamburg allow us to calculate the scattering curves directly from the PDB codes of these crystal structures, and you see that the curve of the trimer fits our data much better than the hexamer. So most probably we have a trimeric sample here. And there is another software tool that allows you to reconstruct the solution structure from the SAXS data and likewise from the SUNS data. You see this as gray spheres superimposed on the trimer structure. And this gives you a quite nice feeling for the uncertainty or capability of determining size and shape of a biomolecule with small angle scattering methods. So here for our sample, we can rule out the hexamer structure. But what can we contribute to the question of the supercomplex that Phycocyanin forms? In usual cyanobacteria, you see the structure on the left side of the slide, which is very complex. You have several rods radiating from a central core. But we studied a system from a particular cyanobacterium called Acariochlorus marina, which is supposed to form a rod-shaped supercomplex of phycocyanins. But it was unclear under which buffer conditions do you really retain such a structure, and how many phycocyanin subunits do you really have in this? So we performed experiments on these phycobiliproteins in phosphate-containing and phosphate-less buffers. You see the phosphate-containing sample as blue points in the slide. And this can be nicely described simply by a cylinder model with a length of about 225 angstrom. So for this buffer condition, we can prove that there is a rod-shaped assembly, a cylinder shape, while if we move to the phosphate-less buffer, then the structure seems to be lost, and the cylinder model still applies, but the height of the cylinder is only 28 angstrom anymore. And in addition, you see that for small scattering vectors Q, there is a huge increase in the signal, which usually indicates unspecific aggregation. So we have more or less the same sample in two different buffer conditions, but the structures we get are completely different. And without a small angle scattering study, you wouldn't have noticed that. You would have compared with the crystal structure that probably does not apply. And with this structural information, you can also explain why the function of the phosphate lacking sample is impaired, because simply the active structure is split into pieces. That is summarized on this slide here. For the phosphate containing sample, we get a cylinder shape that contains 8 subunits. while for the phosphate-less sample, we get non-functioning materials which are split into trimeric phycocyanin subunits and which show unspecific aggregation. Now with that knowledge, we can move to the more complex situation of membrane proteins. And here I show you the structure of photosystem 2 and the structure reconstituted from a small angle experiment as gray spheres superimposed on each other. And you see they seem to nicely fit, while the sphere structure is a bit expanded. So it might be that in solution this complex is a little bit larger. But after my introduction, you may miss the detergent belt around this membrane protein. And why we don't see it, the reason is that we have a so-called contrast matching technique here, which is only available in small angle neutron scattering. And the principle of this is outlined on this slide. You see we have a 3-component system, the protein in blue in the center, surrounded by an orange detergent belt, and this again surrounded by the solvent. And particularly using neutrons, we have the possibility of contrast variation when we use or when we vary the content of D2O and H2O in our solvent, because neutrons are sensitive for isotopes. They study nuclei, and this means we can we can vary the scattering properties of the solvent, and we find so-called match points where the scattering property of the solvent matches that of the detergent belt, while in the second case the scattering properties of the detergent are not matched. So we can do small angle neutron scattering under matching and non-matching conditions for the detergent belt. And this slide shows you the differences in the results that you get and in the data. In the first case, with 5% D2O in the solvent, we match the hydrophobic chains of the detergent molecules, which means we selectively see the protein, while in the second experiment with 100% D2O or with small angle X-ray scattering, we see the envelope of the protein with its detergent belt, and we can model a monomolecular belt of detergent molecules around the photosystem. Please note that the protein with detergent belt is the top view, while the protein selectively measured is the side. So this is a bit probably. But to summarize this subpart, we can measure the photosystem 2 solution structure by small angle neutron scattering with contrast variation, which is very close to the X-ray structure, but seems to be a bit expanded. And especially we can detect a monomolecular detergent layer around the molecule when we use non-matching conditions. So to move to the last example for small angle scattering, the light-induced structures, here we study a protein where only the dark ground state is crystallized, but if the molecule has to become functional, you have to activate it with light, and then its 2 domains seem to split before they can bind to Phycobiliproteins. And we are interested in this first step here that can be induced with 480 nanometer blue light. This means we have to interface this light excitation with the small angle experiment here shown for an instrument at MLZ München in Germany. where you see our illuminated sample container, and it's not sufficient only to illuminate, but you really have to make sure that your sample is really converted more or less completely to the active state. And you see this here in the right lower corner, where an unilluminated part of the sample remains orange as in the ground state, and the illuminated part becomes red. Then we get really two distinct small angle curves for the dark and for the illuminated sample. And the dark sample nicely fits with the dimeric ground state crystal structure, while the active state structure appears to be expanded. And you can model the separation of the two domains with software tools available from the Dimitri Svergun's group at DAISY. So to summarize this part, we managed to convert OCT to its active state under illumination, and we can see a domain separation with this experiment, so we can well characterize the active state. Summarize again, for the small angle part, we can check oligomerization and aggregation. We can test complex formation from systems with known crystal structures. We can determine solution structures at physiological conditions, and even follow conformational changes on external triggers. The second shorter part is about molecular dynamics, because once we see these beautiful pictures from crystal structures, one often forgets that the proteins are rather flexible entities. And to illustrate that, I want to show you a molecular dynamic simulation that I can show you courtesy of Dr. Byers from University of California. And this movie nicely shows you that a protein is a very dynamic entity, and smaller residues are moving on picosecond timescale. We have measured that for photosystem 2 and expressed this flexibility in terms of a mean square displacement. You see here in this slide, at low temperature, this displacement is rather small, but towards physiological temperatures, it increases a lot. So especially for physiological processes, we have to keep in mind that flexibility is important, but we do not see so much flexibility in the dry state. So hydration of the molecule seems to play a role, and for that, I switch on the video once more and tell you that the dark gray is the protein itself, while the light gray red molecules are the water molecules that are wiggling on the surface of the protein and are initiating the motions of the protein. And you see that with our neutron spectroscopy, you can measure these displacements. Why is this important? Here you see the curve from neutron spectroscopy superimposed with a measurement of electron transfer efficiency, and you see that the two correlate. So there are functional processes in photosystem 2 that seem to require flexibility, and there are several structural models that show us or that give us an idea which smaller conformational changes in the photosystem 2 may require conformational flexibility. And very finally, I show you that we are setting up experiments for time result studies of dynamics of biomolecules, here shown for the example of bacteriorhodopsin, where you can initiate its function also by a light quantum. Then it undergoes a photocycle through several structural intermediates. And if you interface a neutron spectroscopy experiment with external illumination, you can, instead of a snapshot of the dynamics, in principle measure a movie through all these functional states. And this is what we have here. So to conclude, subsection probe neutron experiments can be performed, but they require a lot of neutron flux and they permit preparation of specific functional states and their characterization, but they highlight the active role of dynamics in functional processes. And with this, I want to thank all co-workers for the particular projects that I have introduced here, and I thank you for your attention.
Thank you very much, Jörg, and we have indeed many questions because of such an insightful presentation. The question is, for your study, did you use the same proposal to get the beam time at synchrotron and at a neutron facility? And if it was in the sequence, did you use the fact that you had the BT in either in SRS or a neutron facility? I hope you understood the question.
Yes, but this, of course, this is a very important practical question, but this varies from project to project. And as we have also seen in the talk before, sometimes it's good if you can perform some of your experiments in a laboratory where you have easy access and you can try out things and you can do feasibility studies. But of course, then it's also important to apply for large-scale facilities because you get higher flux. You can study samples with smaller amounts and smaller concentrations. And especially because I feel an interest in combining SANS and SAXS from this question, there are centers like ILL and ESRF who provide the opportunity to apply for both SANS and SAXS beam time at the same time. You submit one proposal and you can pick an option, and once your SANS proposal is accepted, you get automatic beam time at ESRF for SAXS experiments. And especially the Photosystem II study was done in this way, and it turned out to be a really nice combination of complementary methods.
Okay, so you did that in a complementary way. The next question is classical, and this is so specific that only you can understand that, I believe. Classical MD potentials may perform poorly when van der Waals interactions are present. How was the MD potential tested?
For this particular movie that I can show you, I cannot even answer this. I would have to direct this question to Doug because this is not my work. I have shown it for illustration purpose. We are at the moment moving into this direction because I think it is a crucial combination to combine MD with the experimental neutron spectroscopy, simply because MD gives you a better idea of a structural assignment of the motions that you see. So I'm sorry that I can answer this question only in general and not in particular.
But definitely we're going to address this question particularly after the conference and find the answer for those who are asking this question. Then the next question is about the team of authors. At the beginning of your presentation, you showed how many people are behind the research from many countries. And the question is, what does it take to be in such teams and to organize such teams? That's a very human question from everyday's perspective. What does it mean to be a part of something and also organizing something?
Yes, I would say that work on such a complex scientific question requires teamwork to begin with. There is almost nothing that an individual person can do by himself or herself. And of course, all these people that you can see who have contributed in many different ways. First, when you apply for beam time to a scattering center, you get a local contact. and this is a scientist that constantly maintains an instrument and upgrades this instrument to the best standard. And without this basic work of this local contact at the scattering center, a scientist who is just a guest there could not do anything. So this is the local contact at the scattering center is, of course, a very important contributor to such a project. And once you combine SANS and SAXS, you already have at least 2 contacts in the centers. Then, of course, we work with people who have all the knowledge on biochemistry, who know how to extract an isolated photosystem from a membrane and from a plant or from a bacterium, which we as physicists couldn't do. And so all such contributors or collaborators you need to successfully perform such a study.
Okay, thank you. And then at the beginning of afternoon session, there was mentioned the huge role of modeling before going to real experiment. Could you evaluate a bit the stage of modeling, how much time it takes, and what's the reality behind the modeling in a research?
Yes, actually that is a 2-stage process, because a collaborator may turn to you and say, we have an interesting protein, it would fit into our field of collaboration. Can we do a study? Can we apply for beam time somewhere? Then it means you have to analyze the system first and, for instance, check out how large will it be? Which structure do you have to expect? And as I mentioned in one of my slides, from a structure that we expect, we can already calculate a model curve, and this defines the experimental conditions that we have to ask for at a particular scattering center. So when we write our proposal, we should already know roughly what size of protein, what shape of protein should we study, or in these studies with external triggers, which changes do we really expect? And then we can define the experimental conditions and ask for experimental conditions in a more elaborate way. And then, of course, once we have done our experiment and we go home with the data to our home institution in Estonia, then the real work starts for us, because then we have to model and find out which shapes, which conformational changes have we really seen in our experiments. And this can take a long time.
Okay, thank you. And then the final question, which is a very, very human type of question. What would be your advice from Tartu to your colleagues in Riga?
That's a very good question. I would say take the opportunity that is offered by such nice events as you have organized and form collaborations. Because as you say, the author lists on our project proves collaboration is the key to everything.
Thank you. Collaboration is the key for everything. That will be definitely one of the, I believe, conclusions of this conference. Thank you, Jörg, very much for this intense Q&A session.